Today's lecture will be on the human brain, and the first part of the lecture will be mostly concentrating on the structures. Some particular terminology we should look at first is anything referring to the anterior portion of the brain. We consider this to be called rostral, or in other words means that it's towards the anterior direction or towards the nose. Any of the structures located in the posterior department, we call this the caudal area, which is meaning towards the tail. So the two distinct features that occur on the surface of the brain are going to be the outer surface of the brain, which is exhibiting these different folds, and these are called gyrus or gyri, or individual folds. And within those folds, we see the shallow depressions between the folds, and they're going to be called sulcus or sulci. So with these sulcuses or sulci, they're a little bit deeper into the cerebrum aspect of the brain. The first area that we see a deeper sulcus is going to be this region called the parieto-occipital, which extends down, and this is simply going to be a deeper sulcus or a groove in the surface of the brain that separates the occipital from the parietal. The next one is going to be the lateral sulcus. It's again a deeper groove. This is going to separate the temporal lobe from the parietal, the occipital, and the frontal lobe. And the main one that we see is called the central sulcus, and this is going to separate the frontal lobe from the sensory area of the parietal lobe. Other structures that we see, as I mentioned, the cerebrum encases all the four different lobes or regions, the cerebellum, which is going to be working in conjunction with the cerebrum, and finally the brainstem. So with the individual parts in each one of these, as I mentioned, the cerebrum is this outer surface of the brain. It has the different sulcuses that help compartmentalize the different lobes. I mentioned the cerebrum, which is going to fine-tune the motor commands coming in from the cerebrum. So the cerebellum is important for fine-tuning motor and sensory information. And in the brainstem, the first area of the brainstem is going to be the midbrain, and then moving more inferior, we see the pons and the medulla. And the diencephalon is going to have two parts. The first one is going to be this thalamus, which is a two-lobed organ that is connected by an interthalamic adhesion. Your thalamus is important for sending sensory information to particular regions of the parietal and receiving motor commands and then processing them to send them through the spinal cord. The hypothalamus is also part of the diencephalon, and it's important for the initiation of hormone production. So when we're looking at organization of neural tissues, we're going to see two components. We're going to see these outer layers of gray matter and usually an inner layer of white matter. We also see a structure called the cerebral nuclei, which are simply clusters of gray matter within inside of the white matter. So we can see these located in the cerebrum. We also see them in the cerebellum and the brainstem, including the medulla and the spinal cord. Each of these gray and white matter regions have different types of neural tissues inside. So just using this gray matter of the cerebrum, this represents the region, this gray matter represents the regions containing neuron cell bodies, the dendrites, which help communicate. At the end of the axon collaterals, the gray matter also has the terminal arborizations and unmyelinated axons. The white matter that you see in all these illustrations are going to be composed of myelinated axons. For example, the corpus callosum located here, This is going to be communicating, these myelinated axons will be communicating between the left and the right hemisphere. We see this in the spinal cord also. The white matter will form a series of axons that run in a track, leading the information up through the brainstem into the cerebrum. And as mentioned before, the cerebral nuclei, these areas, these dark shaded gray matter areas, these are just clusters of gray matter within the white matter, and they're usually just neuron cell bodies that are located in these distinct clusters. So the next part is to look at the cranial meninges, or the protection of the support. There's 3 connective tissue layers of the cranial meninges. Going from deep to superficial, the deepest one will be closest to the brain, called the pia mater, and then moving more superficial, we'll have the arachnoid matter, and then finally the dura mater. Their job is the soft tissue separates the soft tissue of the brain. So the gray matter, it separates the soft tissue of the gray matter from the cranium. It encloses or protects blood vessels. So we can see here in the subarachnoid space that we have a series of arteries and veins that are closely running next to the pia mater. And we also circulate cerebral spinal fluid. So in the subarachnoid space, we see the cerebral spinal fluid, which will eventually lead out into the venous system and then back towards the heart. So we'll begin the discussion first with the pia mater. So the pia mater is this purple shaded region right on top or adhering to the gray matter, and this connective tissue is called an areolar connective tissue. It's highly vascularized. You can see where we see the arteries and veins running right next to it, and this is important because we're dealing with the gray matter of the cerebrum. Moving up, it will be the arachnoid matter. So within the arachnoid matter, we're going to have a couple of different areas to talk about. And the first one is going to be the arachnoid trabeculae. And the arachnoid trabeculae is this white spider web structure, which is basically composed of collagen and elastic fibers. And this is going to be important to maintain the opening for cerebral spinal fluid to eventually flow through the villi and into the venous system. The subarachnoid space, the arachnoid trabeculae is holding open, so this region from here to the top of the pia mater, this region is considered to be the subarachnoid space. And then we also have a potential space, and the subdural space is going to be, if we would peel back from the upper surface of the subarachnoid space, between there and the meningeal layer, we call this the subdural space. And this subdural space is what's considered to be a potential space. And if a hematoma would occur, or an abnormal bleed in the brain would occur on this region of the subdural space would then fill up with the fluid, so putting the pressure more towards the outside rather than compressing onto the gray matter of the brain. And finally, the last space, the most superficial, this is the hardest one, or the most protective layer. It's composed of dense irregular connective tissue, and it'll have 2 layers. The innermost layer is going to be this one. This innermost layer is called the meningeal layer, and the meningeal layer is important. It's deeper than the periosteal layer, and it's going to be closely associated with the arachnoid matter. And then the other layer that we have is the periosteal layer. It forms the portions of forms the periosteum on the inner surface of the cranium. So the periosteum is that dense irregular connective tissue, and we see this adhering to the base of the cranium. Within this layer also, we have another potential space. So between the periosteal layer and the base of the cranium, this epidural space, this also contains arteries and veins that help nourish all three of the meningeal layers and the bones of the cranium. Again, it's not a real space, it's just a potential space in case blood flow would need to increase in that surface. The other structure to point out, the arachnoid villus, these are just extensions that occur inside from the arachnoid or subarachnoid space into the dural venous sinus. This is where we're going to see excessive cerebral spinal fluid dropping into the venous system and exit eventually to the heart. When we're looking at the cranial dural septa, these septa are basically helping to compartmentalize or form some structure between all the working aspects. So the first one we see is the falx cerebri. You can see it's shaded in white. So it's a connective tissue. It's the largest one that we have. It goes along the mid-sagittal plane, and it's going to basically help separate the superior sagittal sinus and the inferior sagittal sinus are going to run along this region. So this is going to be the largest one. This connective tissue also helps support all the different sinuses, so the straight sinus or the transverse sinuses. So it just maintains a little bit more structure in the brain. The tentorium cerebelli, this is going to separate this one right here is going to separate the occipital lobe from the temporal lobe. It also separates the lobe of the cerebrum, so this area here, from the cerebellum. And we also see the transverse sinus is going to be running along the posterior border of the tentorium cerebelli. The next one is going to be the falx cerebelli. So we're more in the inferior portion. The falx cerebelli is going to divide the left and the right cerebral hemispheres. So this is going to cut all the way through the brain. What we see here is the occipital sinus. The sinus is basically a vein, and it'll be running along the posterior vertical border of this falx cerebelli. And finally, the diaphragma cellae. The diaphragma cellae is the smallest one, and it has a small opening that allows the infundibulum, which attaches basically the hypothalamus, which would be located here, to the pituitary gland. And the infundibulum would be the tube that runs from the hypothalamus to the pituitary. And this will be important for hormone production. So there are 4 ventricles of the brain, and the 4 ventricles of the brain are going to contain the cerebral spinal fluid. So we have a lateral ventricle, we have the 3rd ventricle, we have a series of cerebral aqueducts, and then of course we have the 4th ventricle. So when we're counting this out, the left and the right would be the lateral ventricles, so 1, 2. The 3rd ventricle, which is located deep, closer to the thalamus, and then the 4th ventricle, which would be located next to the cerebellum. So the first structure that we would look at in the lateral ventricles is going to be this intervertebral foramen that's going to connect the cerebral spinal fluid from the left and the right ventricles. On the 3rd ventricle down here Um, we see this within the diencephalon, and the lateral ventricles will then be sending again through the intraventricular foramen. The cerebral spinal fluid would be flowing down into the third ventricle. It would then go through the cerebral aqueduct, and through the cerebral aqueduct, this is contained between the third and the fourth ventricle. And then eventually it'll come into the fourth ventricle where it narrows at the inferior end, and it's going to either go through the lateral aperture, which is an opening, median aperture, or down through the spinal cord. So the way the cerebral spinal fluid is produced is through these epididymal cells that are closely related to a capillary system, or the end of an artery or a capillary. And this connection is going to allow for the body to pull out blood plasma from the capillary, and it'll be pulled in from the epididymal cells, and then it'll be able to enter into the ventricle. You can see the epididymal cells have a series of microvilli or cilia located on the outside. So the process would be, as the red blood flow is coming through, or red blood cells or the erythrocytes, we would see the plasma being pulled out, the epididymal cells then transferring that into what we consider to be the cerebral spinal fluid. It would then go into the ventricles, go through all the ventricles, and then eventually into the venous system. So if we look at a section of the epididymal cells, if we put these together in an area such as in the lateral ventricles, as this picture is showing, then we would call this the choroid plexus. And that just simply means that we have a region of epididymal cells that are producing cerebral spinal fluid and pulling it into the ventricles for circulation. Their job is, movement buffer. It helps the environmental stability, such as nutrients, waste, chemical fluctuations. So there's a lot of important functions that occur with cerebral spinal fluid. So this chart's going to take us through the beginning. We'll start here at number 1. So number 1, we see that the cerebral spinal fluid is produced in the lateral ventricles, and it's going to be produced by the choroid plexus, or a series of epididymal cells that are pulling the plasma from the blood and making cerebral spinal fluid. So the next part is then to travel, flows from the lateral ventricles and eventually into the third ventricle, and it's going to go through this narrow constriction right here, and it's eventually going to reach number 2, which is this region here, which is considered to be the third ventricles. From this part, then we're going to see it traveling through the cerebral aqueduct, and as it goes through the cerebral aqueduct, then we're eventually going to get to the fourth ventricle. So in number 3, we see that the cerebral spinal fluid in the fourth ventricle is then going to pass through these lateral apertures and a single median aperture into what we consider to be the subarachnoid space. So we're going to see it come through this median aperture and a lateral aperture, and it is going to come into this region of the subarachnoid space, and it's going to go in 2 directions. It's going to come up towards the cerebellum, and then after it drops through the lateral apertures, it gets in the subarachnoid space, and then it's going to make its way into the front or the anterior portion of the subarachnoid space. It's going to come around the pituitary gland, and then eventually it's going to make its way completely around the brain to number 4. So as it's flowing through the subarachnoid space, we say that it's providing buoyancy for the cerebrum, the cerebellum, the brainstem, and also the pituitary. It'll continue to travel, and then this illustration will show a bigger view, and I'll switch over. So when we get to number 5, we're traveling in this upper view. The cerebral spinal fluid is traveling through the subarachnoid space, and then eventually what it's going to be doing is making its way through the arachnoid matter into the arachnoid villi. And this excessive cerebral spinal fluid flows into the arachnoid villi, and then it's going to drain into venous circulation through an area called the dural venous sinus. So now we're back at the lower picture. We're in the darker blue now, and we're traveling through the dural venous sinus. And basically, the greater the pressure means that ensures that the cerebral spinal fluid will be moving into the dural venous sinus without permitting any venous blood flow from entering into this region. So the cerebral spinal fluid, in essence, is traveling under a lot of pressure, and it's pushing through the one-way valve, but it's not allowing the venous blood to enter into the subarachnoid space. And we discussed a little bit, quick note, with the cerebral spinal fluid production, we produce about 500 milliliters per day. And again, we find cerebral spinal fluid being produced in the choroid plexuses. And then finally, we talked a lot about this. This is a glial cell of the central nervous system. It's called the astrocyte. We mentioned a little bit about this in the nervous system discussion. But the astrocytes are basically the gatekeepers. They have these structures called perivascular feet, these right here, which are attaching to the capillaries that are coming around the brain in the subarachnoid space. And within this, what we see is the lipid solid compounds can enter freely. So nicotine, alcohol, anesthetics are permitted to pass through this blood-brain barrier. And other items, the astrocyte would have to change the endothelial lining. And what we see is that there's a tight junction between each one of the endothelial lining, and we have a continuous basement membrane. The erythrocyte would be located on the inside, and the astrocyte will dictate the permeability principles and will allow what particular ions need to come into the interstitial fluid of the brain. So we do see a reduction of the blood-brain barrier. So we don't see astrocytes in 3 areas. The first area we don't see astrocytes in is the choroid plexus. In the choroid plexus, we don't see astrocytes because the capillaries of the choroid plexus must be permeable to produce the cerebral spinal fluid. We also don't see this located in the hypothalamus or in the pineal glands. So both the hypothalamus and the pineal glands produce hormones that need to have easy access to blood. So we are essentially just making sure to protect the neurons from any chemicals, waste, hormones, or high or low levels of ion levels that are building up in interstitial fluid.